Hi and welcome everyone to our BIS103 map. This one is part 5. So this one will actually be our near complete map. And what I want to do in this part of our map is to give you a big picture overview of what we have achieved, all the pathways that we have discussed in our lectures 1 all the way through lecture 16, and give you a foundation for this knowledge of pathways, how they cooperate, how they intersect, as we're discussing today in lecture 17, metabolic integration, and we're looking at how these pathways work with each other, in conjunction with each other, under different cellular and organismal conditions. So what do we achieve so far this quarter? Right? We started out talking about our carbohydrates, our glucose, how we can break it down to pyruvate in our glycolytic pathway here. Then we talked under aerobic conditions about how we can use our PDH reaction, right, to convert pyruvate into acetyl-CoA, one of our key central metabolites in energy metabolism. We had talked about a number of other food resources, other metabolites that are energy rich that we can bring into these catabolic pathways to ultimately make ATP, right? One being here our carbohydrate storage, glycogen or starch, how we can break it down into glucose 6-phosphate or glucose. We had talked about some other carbohydrates, right? Our disaccharides, some other monosaccharides, and how we can feed those into glycolysis for breakdown, for energy metabolism. And then we had moved on. We had talked about other energy resources, right? Our lipids and fats. We had talked about how we can use hydrolysis of lipids and beta oxidation of fatty acids to produce acetyl-CoA. We had also talked now about our amino acids more recently, right? How we can break down proteins through hydrolysis, and then amino acids and their different breakdown pathways into either glycolytic intermediates or TCA cycle intermediates, the majority of which ending up here in acetyl-CoA, some other TCA cycle intermediates, right? That brings us to our central wheel of metabolism, our TCA cycle, where we're using acetyl-CoA, we're decarboxylating it, and in the process, right, shuffling the electrons into our electron carriers, NADH and FADH2. And then we had said that the outcome of the TCA cycle can be twofold. It can either be used for the electron transport chain, the ETC, or we can use some of the intermediates also as biosynthetic precursors. First, we then had focused on making energy, right? We had used now all the electron carriers coming out of the TCA cycle, as well as your PDH reaction, your reaction 6 out of glycolysis, all the other oxidation reduction reactions that generate reduced cofactors. Right? We could use those through the ETC to generate this proton motive force that then was driving our ATP synthase, and we finally made ATP. So we looked at all these catabolic pathways for the production of chemical energy in form of ATP. We also looked at what is happening if we actually don't have oxygen, right? If the PDH reaction is not running. We had talked about lactate formation and ethanol fermentation, looking at other pathways that allow us to regenerate our cofactors in the absence of oxygen. And then we had started to turn things around and looked at some of the anabolic pathways, right? How can we generate other metabolites? And we had talked about these TCA refill reactions, our anaplerotic reactions, how we can refill the TCA cycle, right? If we're using it for the production of biosynthetic precursors for other pathways, we have to refill the cycle so we can still run it for the purpose of EDC. So these were our anaplerotic reactions. Right, we had talked about GNG, or gluconeogenesis, and how we can actually use, for example, lactate, amino acids, other metabolites, and can use our gluconeogenesis running essentially, right, glycolysis in reverse, with some enzymes being unique to overcome the irreversible reactions of glycolysis, but we could use those to generate glucose 6-phosphate from non-carbohydrate metabolites. We had talked about this very specific, right, liver phosphatase that can dephosphorate glucose 6-phosphate and realize free glucose. Again, right, this was specifically in the liver because the job of the liver was to release free glucose into the bloodstream to support the entire organism. We look at this phosphatase one more time actually in lecture 17 today. Then we had talked about, well, how can we go further from glucose, right? How we can actually make glycogen from glucose or glucose 6-phosphate. Again, our anabolic pathways for storage purposes. We had done the same, right? Looking at how we can use acetyl-CoA in the reverse reaction of beta oxidation, looking at how we can make fatty acids. We had done the same for our amino acids, right? We had looked at how we can use TCA cycle intermediates and glycolytic intermediates and bring about the biosynthesis of amino acids with the same kind of chemical logic, right? We looked at some of the reverse reactions, and we looked at some unique reactions that helped us to overcome the irreversible reactions of amino acid breakdown to actually build amino acids from other precursors. And we'd also talked about our PPP, right? Our pentose phosphate pathway and all its many important functions, right? As it's intersecting with glucose 6-phosphate as a substrate to the pathway, how it helps us to, for example, generate or catabolize some of the unusual 4, 5, and 7 carbon sugars, important, for example, in nucleotide biosynthesis, but also, right, our C4 sugars, for example, are a precursor for proteins and amino acids. And importantly, right, our NADPH, we had seen it in a number of reactions where it was used as a cofactor, one important one being fatty acid synthesis, for example. And we had a few sort of side topics on top where we had talked about ketone bodies, how these are a pathway that can happen under certain cellular conditions, especially when you're running out of glucose, right? You can siphon off acetyl-CoA to produce ketone bodies, which help you to overcome possible limitations in your cofactor by regenerating the free CoA, but also using ketone bodies now as a transportable form of acetyl-CoA to use for generating chemical energy elsewhere in the body, especially in the brain. We hadn't really talked about how we actually do this, but we had talked about their general purpose. And then one more add-on, right? We had talked about this unusual glyoxylate cycle that animals don't have, but plants, fungi, and select bacteria have it, that allows these organisms something that animals cannot do, and that is the net production of glucose 6-phosphate from acetyl-CoA using this bypass cycle that bypasses the decarboxylation reactions of the TCA cycle. So we have achieved a ton, right? What I want you to focus on is to sort of keep in mind these really broad picture pathways and how they cooperate with each other, how they run in opposing reactions under different conditions. And this, I hope, will help you to have a good foundation to understand and follow all these different metabolic integrations that we want to discuss in lecture 17.